Hi everyone, don't forget to subscribe to my channel and let's see what we have today. Let's review what we talked in the previous video about DNA structure. As we learned, DNA is made up of nitrogenous bases, deoxyribose sugar, and phosphodiester bound. And these components are bounded together to give us nucleotide, and this nucleotide is bounded together to give us the DNA double strands. And they are antipolar and complementary, where one strand starts from 5 to 3 and the other is a complement and starts from 3 to 5. 3 and 5 refer to the number of carbon atoms in deoxyribose sugar to which a phosphate group bound. Bacteria multiply by binary fission, resulting in the production of two daughter cells from parent cell. So, each daughter cells receive an identical copy of DNA. So, how did this genetic material pass from the parent to daughter? Actually, through a process called replication, in which a DNA double strand is copied to produce two identical DNA molecules, and this process occurs in the eukaryotic nucleus and in the cytoplasm of the prokaryotic cell. But most importantly, this process takes place before the cell division. For replication process to be accurate and rapid, it must go through several stages. As we said, the DNA is highly coiled, and in order to replicate, it has to relax firstly and separate the two strands, and this the first stage in the replication process. Secondly, each strand may serve as a template for DNA synthesis of new strands. Lastly, termination of replication and releasing two identical chromosomes, one for each daughter cell. Alright, now let's get to the core of the process. Imagine we had a big company and it had 4 partners, each with their own personalities, and this company wanted to open a new branch somewhere, so they decided to meet with each other. Well, let's see what happened at that meeting. One of the partners, called Helicase, he has a nice, quiet personality that make everyone meets him relax. So Helicase is going to relax the double strand of DNA in an area called origin of replication or replication fork. This fork is two bidirectional, in which each fork moves through the parent DNA in an opposite directions. But let's not forget the helicase has a supporting team. One of the members of this team is called SSP proteins, and these proteins bind with the strand of DNA after separate to keep them from rebinding. Also, they are ribosome, which are cooperative protein complex, in addition to origin recognition complex proteins, which have role in determining the site of replication fork on the strands. The CEO of the company is named DNA Polymerase, and he has a tough personality. He has a one-way mind, and he always needs to be guided and informed about all the meetings. And it's the third character's job, RNA primase. DNA polymerase cannot start working without primase guiding, so the primase put a little bit of RNA primer on the DNA strand so the polymerase can start building the new strand. But we have a little problem. As we said, DNA polymerase has one way of thinking and it can only build from 5 to 3. So what's the solution? And we have another DNA strand that starts from 3 to 5. You are feeling confused? Well, let's make this little clear. So, as we said before, DNA is made up of two strands, one from 5 to 3 and one from 3 to 5. Well, when the process of cloning starts, those strands break apart to clone themselves. In the original DNA strand that goes from 3 to 5, DNA polymerase will be built from 5 to 3 because, as we know from the properties of DNA, it's antipolar and it's basically the direction that DNA polymerase knows. So we don't have a problem with that strand, and that's why this strand is called leading strand. Unlike leading strand, the original second strand of DNA, which starts from 5 to 3, has a structural problem because DNA polymerase can only be built from 5 to 3, and the new strand must start from 3 to 5. But there is a solution. Primer bind to the strand to guide DNA polymerase to start building, then it will stop until another primer reattaches to the strand and continue the process. This strand called lagging strand. Unlike to what people have been saying, DNA polymerase changes its direction, but in fact it doesn't change direction. It's driven by the primer. This process makes a space called okazaki fragment, and to solve these gaps, we call on the peaceful fourth character, which called ligase, to connect them to each other and fix any errors that may exist in the process. So in the end, we have a new two double strands, which are called semi-conservative because contain one copy from original DNA and the other is new strand. In bacterial replication, time is vary. For example, E. coli or Escherichia coli takes approximately 20 to 40 minutes, and so far it varies from strains to strains depending on the environment, such as availability of food or the presence of toxins such as antimicrobial agent. The next video is going to be very interesting. It's about gene expression, which is the processing of information encoded in genetic material that result in production of RNA and protein. So stay close.